The group of scientists of Molecular and Radiation Genetics Department of DLNP JINR studies Drosofila melanogaster to understand changes in DNA under the influence of ionizing radiation and to search for new elements regulating the work of various genes. In the structural part of eukaryotic genes, coding sequences of nucleotides exons are interrupted by non-coding sequences introns. The gene activity can be regulated by enhancers, which increase the activity of a target gene, silencers, which on the contrary reduce the activity of a gene until its complete shutdown, and insulators, which isolate a gene from silencers and enhancers action. All these regulatory elements can be located in any position in a genome, including introns and on other chromosomes in relation to a gene. An ionized molecule releases energy which causes chemical bonds breaking inside and between nucleotides and damage of the original DNA structure. As a result, a mutation occurs. There are different types of mutations: deletion, the loss of some part of DNA; insertion, DNA fragment insertion; inversion, when DNA is 180 degrees flipped; and base pair substitution. All these types of mutations occur under the action of ionizing radiation and lead to disruption of gene activity and as a result to a change in Drosofila features. Radiation-induced mutations can affect both the gene itself and the regulatory elements that control it. The mutants can be visually detected by inspection of various Drosofila features. Unique JINR facilities provide a remarkable opportunity to obtain all types of mutations in any position in a genome. DNA of the obtained mutants is sequenced. Sequencing is the method of determination the precise order of nucleotides within a DNA molecule, which allows to detect mutations which are the cause of hereditary diseases, hereditary predispositions, or particular features of the organism. The DNA structure is determined by the extraction of DNA, its special processing, and then obtaining information by means of a special device called the sequencer. Sequencing of these mutations makes it possible to determine their type and frequency of occurrence, and the comparison of the changes in the functioning of the organism makes it possible to identify unknown regulatory elements which control the work of the gene, or find new genes involved in certain biological processes. About 60% of Drosofila and human genes are similar. For example, 2/3 of oncogenes in humans have functional orthologs in the fruit fly. Therefore, understanding the mechanisms of gene regulation and their damage under radiation allow to assume how these processes occur in humans and learn how to manage them.